So I recently cut up some brains. Don't worry, totally legal. It's for science. Now I want to mark all the brain cells that activate during the experience of pain so that we can target and potentially shut down these cells as a way to manage chronic pain. First, I know I only want to look at one particular area, so I have this atlas that tells me exactly what the slices I want should look like. So I select out the ones I want from all of my 14 mouse brains and get them ready for staining so that we can, again, tag these pain activated cells. And the staining process we use is actually quite cool. It's a very good example of scientists using what our bodies already make to help unlock its mysteries. So your body naturally makes molecules called antibodies that help find and fight germs. You can think of them as like tiny keys that only fit particular disease targets. Like there's likely a very specific key for the flu molecules that are floating around in your body right now, just waiting for the flu to come around so that it can fight it off. So scientists realized, we can use the same idea in the lab. What if we took antibody keys that are made to specifically tag and stick to the brain cells that are recently active because they make a particular molecule called FOS. So we create this key for FOS, or I didn't make it, I purchased this online, but we make it so that it can tag all the cells that are responding to this particular painful experience. So this is what I've added here. And tomorrow I'm gonna use a different key that glows a particular color. In this case, it's gonna be purple, and it's gonna recognize this first key so that we can see all these pain activated cells under the microscope. So I have my brain slices floating in these little wells, and instead of taking out each one and moving it from plate to plate because you have to put it in all these different solutions, I'm using this white net well thing that allows you to just easily transfer all of the brain cells from one plate to the next. And I'm putting them on this shaker at room temperature overnight. Then tomorrow we'll finish up the staining and do a microscope reveal. Thanks for watching.